Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Carl Wu from Johns Hopkins University on the show. Please let me briefly introduce you to our audience. You received your PhD in biology at Harvard University and did your postdoctoral work as a Harvard Junior Fellow. You then were a principal investigator at the National Cancer Institute for 30 years. You then joined Johns Hopkins University in 2016 as Bloomberg Distinguished Professor with appointments in the Department of Biology and the Department of Molecular Biology and Genetics. You've had a long career in molecular biology, particularly in chromatin biology. What initially sparked your interest in this field and how has it evolved over the years?
I think my interest really comes from reading about gene regulation initially, first in bacteria, stories about the lac repressor and the lac operator, which were published in Scientific American when I was an undergraduate in college. And then wondering about, how does this translate to animals and plants? I came across the work of Sally Elgin, who was my PhD thesis advisor, but this was before I joined her lab, that I had read some of her reviews about how chromatin structure was related to gene expression, and in particular, what in those days, this was in the 1970s, the early 1970s, that there were these things called non-histone proteins. So I thought, well, maybe the lac repressor for animals existed in these non-histone proteins, and that's how I basically ended up working as a PhD student in Sally Elgin's lab. And of course, that immediately brought me into the whole chromatin arena.
Yeah. So let's talk about your science that centers around eukaryotic chromatin structure and function, biochemistry of nucleosome remodeling and gene transcription, and also then later on live cell single molecule imaging, that's the right term, of histones and nuclear protein dynamics. So when we go back to the days that you just mentioned, your first paper in Cell was published in 1979, so you can look back on a long research career. We won't be able to talk about everything you did, but we'll probably only be able to scratch the surface. So let's start. In the beginning of your career, you worked on DNase hypersensitive sites in metazoan chromatin and mapped the first DNase hypersensitive sites to upstream promoter elements for Drosophila heat shock genes. Can you talk about how you about your discovery of DNase hypersensitive sites and how this changed our understanding of chromatin accessibility and gene regulations?
So that was a complete accident. My thesis project was to follow a really influential paper by Weintraub and Grudin, in which they used DNase 1, which was a probe of chromatin architecture and accessibility that had been used previously by others. But what Grudin and Weintraub did in their paper was to show that the globin gene in red cells were basically more sensitive to cleavage than other parts of chromatin. And so I thought that would be a very cool approach to probe the structure of the heat shock genes, which was, I think at the time, just barely cloned by people in Matt Nesselson's lab, which was just down the hall from us at Harvard. And the only wrinkle I added to that experiment was at the influence of Paul Doty, who was one of my professors and a physical chemist of nucleic acids. Paul, did everything by studying kinetics, and a lot of things by studying kinetics. And So what I wanted to do was to look at the kinetics of digestion by DNase 1 over time. And instead of, in those days, doing solution hybridization, nucleic acid hybridization, so-called cut curves, which I don't think anybody remembers here now, but it's basically a way of measuring the amount of nucleic acids that one has that is complementary to your probe, which is the way the globin experiments were done. I was really excited and fascinated by a paper that I'm sure everybody knows, they should. Southern, 1975, Journal of Molecular Biology. Southern blotting was to transfer DNA fragments from a gel and then transfer it onto nitrocellulose membrane and then hybridize with your radioactive probe the digestion characteristics of any fragment. So I basically did that experiment. I cut Drosophila nuclear chromatin with DNase 1 and ran the products on a agarose gel, blotted it, and probed with the heat shock gene, which was radioactively labeled. And shockingly, we got, instead of a smear, is what one would have expected, if DNase 1 was cleaving randomly throughout the genome, we got fragments, bands. And I actually thought maybe I made a mistake. Instead of using DNase 1, I took a tube of restriction enzyme and threw it onto the chromatin. But indeed, it was not restriction enzymes because the bands were not quite as sharp, but quite fuzzy, but nonetheless clearly discernible over the background smear. So clearly that meant that DNase 1 was kind of behaving like a restriction enzyme, kind of. And there must be local sites where the DNA was hypersensitive to cleavage. Otherwise, we wouldn't see those bands. And so that really, I think, that was reported first I think publicly in 1978, I believe, at the Gordon Conference on chromatin structure. Basically my entire career, you might say, has been trying to figure out what does it all mean? What is a DNase hypersensitive site? And now we know, of course, from our work, from many other people's work, that there are actually gaps in the chromatin, gaps in the nucleosome array, which are nucleosome-free. People call them nucleosome-free regions. And they're created by sort of a combination of the activities of a whole class of enzymes that we and others discovered, called chromatin remodeling enzymes, that use the energy of ATP hydrolysis to mobilize histones on the DNA, mobilize nucleosomes, and either move them aside, reposition the nucleosomes, or in some cases, and under certain conditions, can even evict the histones from the DNA. These remodeling enzymes sometimes will work with sequence-specific transcription factors that recruit them to specific targets, like to the heat shock promoter. There's a protein called the GAGA factor, GAG is the motif that this protein recognizes. And the GAGA factor recruits one of the remodeling enzymes that we study called NURF. It's an I-switch ATPase.
Yeah, maybe before we come into this factor, maybe I can jump in and ask another question.
Yeah.
Because what we are probing now is also like we're using ATAC-seq and other methods to probe similar or maybe even the same thing. So how do these methods compare to those DNase hypersensitive sites you identified? And how are you looking at those methods that were then developed after that?
Yeah, so, I'm totally amazed that there's genome-wide mapping of DNase hypersensitive sites. I never thought that this would be possible, but we had actually randomly sampled about half a dozen genes or loci in Drosophila, and everywhere we looked, we would see hypersensitive sites in the vicinity of the gene. Okay, so a random sampling always brought up DNase hypersensitive sites, but genome-wide, now we know for certain over the entire genome that, in fact, these sites exist, generally at regulatory DNA elements, enhancers, promoters, insulators. So that to me was a great advance. I would say, industrialization of DNase 1 hypersensitive site mapping. And then more recently, as you mentioned, ATAC-seq, which is a transposase-based approach to probing chromosome accessibility, makes it even easier to do. And I love it. I think that this basically has turned into a sort of a fundamental tool of chromatin biology. Anybody can do it, which is, I think, great. But what it tells you is really that, by and large, the original sort of concept that there were gaps in nucleosome arrays, which were hypersensitive cleavage, is in fact, very widespread. And you can not only, map the positions of functional DNA elements like promoters and enhancers, but you can use it to look at tissue specificity. You can look at pathological states. So it's turned out to be a really useful tool. And you might expect that now because you know that these are, in fact, the key signatures of chromatin that are involved in gene expression. And so, in fact, when genes are misexpressed or deranged in their level of expression, you should be able to see some effect on DNase or ATAC-seq sensitivities. Mm-hmm. That's just part of the story, of course. I mean, there are other genome-wide methods for probing footprinting proteins on DNA, transcription factors, and so forth.
Yeah, what you then wanted to find the root cause for those DNase hypersensitivity and those hypersensitivity sites. And you developed an assay to identify transcription factor footprints on chromatin. So what were the major...
Right, so that was the sort of the hypothesis that the underlying mechanism must have come from some combination of transcription factors that would footprint or bind to targets and displace. The original hypothesis was that these transcription factors could, maybe alone or in combination with others, displace histones. And so that turned out to be partly true, but not completely, because the next shock was that, in fact, there's this class of enzymes that use ATP hydrolysis to move nucleosomes around. We didn't expect to see something like this. So that turned out to be actually quite a productive line of research that continues both in our lab and in other labs.
Yes. What you then found is like the heat shock transcription factor 1. And you were able to purify that and then to probably study it. So what did you learn from this about the function of HSF1 as like heat shock transcription factor 1?
So the heat shock locus is responsive to temperature rise. A brief temperature rise in the environment will trigger the activity and the induction of this factor, and it's universally conserved from in all eukaryotes. So by footprinting, we found this activity, which later turned out to be the heat shock transcription factor HSF1. This is the master regulator of the temperature response in cells. And so we just did, so we used that footprinting on chromatin initially as an assay, which is kind of laborious because you're assaying hundreds of fractions because you're trying to make extracts and put the extracts on columns and get sort of activity profiles for this protein. But eventually, and there were more efficient assays, DNA binding assays, EMSA assays, gel shift, mobility shift assays. We ended up purifying this protein, sequencing it, and so on. But this is not the protein that actually is the chief sort of chromatin remodeler per se, in the sense that there's another protein that sort of paves the way for the heat shock transcription factor to do its job. In Drosophila, this is the protein called the GAGA factor, which recruits chromatin remodelers. Then when you have now a hypersensitive site, an accessible site, a nucleosome-free region, on that promoter, the heat shock transcription factor takes advantage of that and binds if more efficiently to that accessible DNA to then trigger the whole cascade of events that leads to the transcription of the heat shock genes. And that cascade of events involves also the assembly of the transcription prenucleation complex, the establishment of a paused state of RNA polymerase II. This is a state that was initially discovered in John Liss's lab. And what the heat shock transcription factor does is cause the release of this paused polymerase and the recruitment of other additional polymerases to give you this burst of transcription. This happens really fast, happens within seconds.
So,
which is part of the reason why we eventually and now have gotten deeply into looking at sort of second scale and sub-second scale kinetics of this whole chromatin remodeling and transcription response.
So you were talking about ATP-dependent chromatin remodeling enzymes earlier, and two people that were in your lab and worked on that were Peter Becker and Toshio Tsukiyama. There were people in your lab to elucidate the molecular basis of chromatin accessibility and the chromatin remodeling enzymes. First, Peter Becker reconstituted nucleosome arrays on heat shock gene plasmids in vitro. So can you talk about this work and what he found there?
Yeah, so Peter's great contribution has been to set up a chromatin assembly system from Drosophila embryos. And he did this on his own initiative because we were trying to get chromatin assembly going on with frog extracts, and there was a period of time it was very hard to get frogs in the U.S. And so sort of in trying to get around this problem, Peter said, "Oh, we have a fly room that's making kilos of Drosophila caviar, Drosophila embryos." And so he basically adapted the frog chromatin assembly extract to Drosophila. And that turned out to be a fantastic approach and a fantastic system, which is used even to this day. This happened, over 30 years ago. But so that allowed us then to chromatinize any piece of DNA that we could get our hands on from the point of view of getting a recombinant DNA plasmid, and put it into Peter's system, and now we ended up with chromatin. It was the nucleosomes were very well spaced and so on. Of course, we didn't know that there were these ATP-dependent activities present in that extract. All we knew was that you needed ATP to create periodically spaced chromatin. And this was actually following the work of Abe Worcel, who had done some really pioneering studies on chromatin assembly in the frog oocyte system. And so without knowing it, we used a system that was going to reveal its secrets to us in the form of chromatin remodeling enzymes.
So that
was Peter's great contribution. He got the system set up. He went back to Germany, to the EMBL, set up his own lab, set up that system again. And the history shows that he's done wonderful things with that extract, and also with the remodeling enzymes that his lab purified and studied in parallel with ours.
Yeah. So what you did, and this was Toshio's work, he discovered the first ATP-dependent chromatin remodeling enzyme, which is NURF. So how did he do it, and what did this mean for the field?
Well, yeah. So Toshi picked up on Peter's project after he left. There was no overlap. Unfortunately, there was no overlap, but Toshi was smart. He picked it up and ran with it. And then the idea was to basically take advantage of the knowledge of this GAGA transcription factor, which other labs, Sally Elgin's lab and other labs, had identified to be present constitutively on the heat shock promoter. And so our experiment was to take that GAGA factor, chromatinize the heat shock gene using Peter Becker's chromatin assembly extract from Drosophila embryos, unpurified, crude extract, add the GAGA factor to that, and see if we could recreate the DNase hypersensitivity at the heat shock promoter. So that was the experiment. That was the plan. And indeed, it was successful. He was able to do this. And the surprise was that you needed ATP. Remember, I said that we initially thought that the transcription factors by themselves could compete against the histones. But in fact, you could chromatinize the plasmid first, you add the transcription factor later. It would work, it would still work, but you had to have ATP in the system. So that meant that there was something in this crude extract that was using ATP to work together with the GAGA transcription factor to create chromatin accessibility. And so that set the stage to purify this activity. And indeed, NURF was the first chromatin remodeling activity that was identified. And it was purified a year later, again, by Toshi in our lab through seven chromatographic steps, completely unbiased purification. Sort of a classic biochemistry approach. And at the end of the day, he ended up with a purified protein complex of 4 subunits, of which one in the paper that accompanied the purification of NURF manuscript, the identification of the I-switch ATPase as a subunit of NURF. And this was done in collaboration with John Temkin, who had generated antibodies against the Drosophila I-switch protein and was encouraging us to use it to probe our fractions. And so we did, and we also sequenced our purified NURF, and lo and behold, the protein sequence, the I-switch ATPase, and the antibody reaction converged on the same subunit. So that was great. And so there were 2 papers in that period. One was the purification of NURF, which was unbiased, and the second was the identification of I-switch ATPase, both by protein sequencing and in parallel, the John Tamkin's antibody. So that was and of course what is I-switch? It turns out I-switch is a family member of the switch-NIF ATPase family that other people had been studying initially by genetics. Yeast switch-NIF is known to be responsible for transcription of a number of genes, and it was proposed to act through chromatin because of genetic behaviors of those mutants. And around the same time as our work, a number of other labs, Craig Peterson's lab in particular, in collaboration with a number of other investigators, I believe it was Jerry Workman as well, purified the yeast switch-NIF complex, and then showed that it too had ability to remodel chromatin. So really, the sort of the convergence of these 2 approaches brought the chromatin remodeling field really smack into transcription, and that then of course between I-switch and switch-NIF highlighted other members of the family, which we identified because the yeast genome had just sort of been sequenced. And so other members of that family basically by sequence homology reveal themselves. The NO80 family, of which SWR1 is a member that exchanges histone H2AZ into chromatin, and the CHD family, and so on. So this is sort of that work, the yeast switch-NIF work, the Drosophila I-switch work, I think laid the foundations of the current field of chromatin remodeling.
So can chromatin remodelers like NURF act on chromatin by themselves, or is there any other factors involved that they would need to act on chromatin?
The NURF does the job by itself. The purified NURF, or now the recombinant NURF, can mobilize what we call histone octamer sliding. They can reposition nucleosomes on DNA by themselves with no help. Mm-hmm. And I think they're the dominant players in the sort of the creation of DNase hypersensitive sites or chromatin accessible sites. Transcription factors help. They can help in several ways, and this is in fact an issue of some debate, current debate, as well as intensive study. In one sense, they might help by acting as a wedge to help the remodeler start its job. That would give it this quality of what has been called a pioneer transcription factor that has been introduced by Ken Zaret's lab. And then they can also, you can imagine, but upon DNA binding, block the translocation of nucleosomes that are being mobilized by chromatin remodeling enzymes. And so in this case, this would be like a barrier against nucleosome encroachment on the gene promoter or enhancer. And there's some evidence, in fact, that the transcription factors can act as barriers to nucleosome translocation, but the evidence that they act as a wedge, that they simultaneously distort. So clearly, when they bind, they have some of these factors can distort nucleosome architecture, sometimes distort them quite substantially. But whether that distortion is causal in the ability of remodelers to do their job in mobilizing the histone octamer is not, to my knowledge, is not been established. And I think that's really a very important question that deserves a lot more attention going forward, which is how do transcription factors, pioneering transcription factors, and remodelers collaborate, if they do at all. What we do know is that for the GAGA factor and now for an increasing number of transcription factors that are involved in this process, there are physical contacts, the domains of the transcription factor that interact with domains of the chromatin remodeler. And so what the presence of these factors, transcription factors, what their presence may help is the recruitment, increasing the local concentration of chromatin remodelers in the vicinity of promoters and enhancers, and therefore allow the remodelers to do their job. But whether, in fact, all three of them, that is, the remodeler, the transcription factor, and the nucleosome, whether all three of them have to sort of work together is really not clear at this point.
You then switched gears a little bit, or maybe it's rather the next logical step, and you focused on the histone variant H2AZ, and this work was published in Science in 2004. There you found that SWER1, which you already mentioned, is the catalytic core of a multi-subunit histone variant exchanger that efficiently replaces conventional histone H2A with histone H2AZ in nucleosome arrays. So SWER1 is not a classical chromatin remodeler, but it's able to exchange nucleosomes. So how does it do this?
Well, we're still trying to find out, but we have learned a lot since that 2004 paper, just 20 years ago. We finally have a paper published just a few weeks ago in Cell, 20 years after the fact, that we have a structure for the SWER1 enzyme. Now, of course, there's been some lower resolution structures and from us in collaboration with colleagues. There's been Andres Leshner's lab is our collaborator on the low resolution structures, where Dale Wigley's lab published some years ago a high resolution structure of the enzyme. In our case, the most recent story relates to the high resolution structure of the enzyme in two conformational states. One in which it is bound to DNA, to nucleosome-free DNA, which is what we call an open conformation, and then another conformation where it captures or is captured by the nucleosome, and that's the closed conformation. How those interactions lead to the eviction of histone H2H to be dimer, one at a time from the histone octamer, is not entirely clear. There's both Wigley's lab and our lab have shown that there's unwrapping of the DNA, but that can't be it. That's not all of it. So there's something else must happen because histones just don't fall off by themselves if you unwrap the DNA at the extent to which they are unwrapped by the enzyme. So we think that the enzyme must give the histone a shove. And what part of the enzyme is touching histone H2H2B and pushing it out of the histone octamer on the nucleosome that is being exchanged is not completely clear to us at the moment, but we're working really hard on it. And hopefully in another few months, we might have something to say. But this must be a combination of both DNA unwrapping and histone ejection, if you want to call it that. That will then leave hexosome intermediate. And that's also in the literature these days. Hexosomes are intermediates of remodeling, not only by the sporulin enzyme, but even for the other enzymes that are involved in mobilization per se. The enzyme basically must kick off histone H2B, and then the H2AZ, which is being held by the other hand of the enzyme has multiple hands, then drops the H2AZ, H2B dimer where the hole, as it were, on the histone octamer has been created by the ejection of H2A, H2B. And so now you have basically histone exchange reaction. I think probably within a year, the fundamentals of that exchange reaction will be well known at the molecular level. Mm-hmm. And that's very exciting. I mean, it's as I said, it's 20 years old, the problem. And it will be interesting to compare how this enzyme does this versus how other members of the switch-NIF and I-switch family mobilize histone octamers, and whether it's going to be fundamentally the same mechanism or an entirely different mechanism in which the ATPase is used for a different purpose, in a different way mechanistically, is not clear at the moment.
Can you talk about the dynamics of the regulation of this replacement of the H2A to H2AZ?
Yes. So we've done a lot of work, and this comes to the live cell imaging, single molecule imaging of SWIR1 and other chromatin remodelers, and the in vitro recapitulation of the reaction at the single molecule level using TURF microscopy, a collaboration with TJ Haas lab. We find that these enzymes bind for only a few seconds in the yeast cell, and then they come off. They're constantly coming on and off, and when they're on, they're only on for a few seconds, as I said, and so they must do their job. And in a few seconds, that's all it needs to eject histone H2H2B and insert H2AZ H2B. And by FRET studies with TJ Haas lab, we actually have very good views of the kinetics of that reaction. It takes about less than 3 seconds to unwrap the DNA, lose one of the histone dimers, and then put in the H2AZ H2B dimer. Less than 3 seconds. And in vivo, that's about how long it takes. 3 to 5 seconds. You will see the enzyme bind to chromatin before it falls off. So our interpretation of those live cell imaging single molecule studies is that when the enzyme is bound stably for a few seconds, then that's when it undergoes histone exchange. So what is quite remarkable is that the in vitro studies of exchange and the in vivo studies of chromatin binding and residence time are very well consistent with each other. So that's really comforting to know that we're looking at the same, the right reaction, as it were. But these enzymes at large will do their jobs also for only a few seconds. Switch-NIF, I-switch, 80, CHD, all within a few seconds. But that's all you need to move nucleosomes. Xiaowei Zhuang's lab showed almost now a decade ago, using the 601 nucleosome positioning sequence, that chromatin remodeling enzymes like the RISC enzyme, which is a switch-NIF subfamily enzyme, can move, histone octamers in the range of 10, 20, 30, 40, 50 base pairs over a period of seconds. So again, all you need is a few seconds of time to shift nucleosomes to create this DNase hypersensitivity. Uh-huh. So we know quite a bit now, actually, about the dynamics of chromatin accessibility. We used to think it was pretty static, but in fact, it's highly dynamic. Even though, from a macroscopic point of view, chromatin accessibility, once you create it, and as long as you have the players sort of in the cell, the accessibility lasts, in some cases, for the life of the organism. Yet, when the enzyme binds, it falls off after a few seconds. Well, you can reconcile these two, the microscopic versus the macroscopic events, because what we have is the enzyme exchanges. So as soon as one enzyme falls off, another member of the same family comes right back on. So macroscopically, the enzymes and the chromatin accessibility is, even though it's under constant motion, the accessibility remains open for days, weeks, months, years.
So what makes it decide? So you said there were like constant contacts to the nucleosome, but what makes it decide to like finally evict it? Is there like another factor or a conformational change, or what? When does it decide to evict the nucleosome?
Well, so that's what was a topic of some debate. I think it's more or less settled, but I think there's certainly a lot more work to be done. So who kicks off the histone H2AZ? And so the H2AZ is incorporated at what is called the so-called +1 nucleosome position, which is just downstream of the transcription start the transcription prenucleation complex, and that's the first obstacle to the so-called PIC that must release RNA polymerase, and polymerase must traverse the +1 nucleosome, move on to the +2, etc., to transcribe. And H2AZ is enriched in the +1 location. And exactly what it's doing, still not totally clear, but it's thought that H2AZ containing nucleosome has different biophysical properties and biochemical properties, certainly biochemical properties, that would facilitate the traversal of RNA polymerase II through the +1 nucleosome. So my bet is that it's polymerase, which is going through the +1, and in so doing, evicting H2AZ. Now, if H2A were there, it would also evict that. And in yeast, H2AZ is not an essential protein. So you can live, although you're not very happy as a yeast, without H2AZ. So polymerase will plow through both H2A and H2AZ containing nucleosomes. And I think and we have some evidence for that published. That's likely to be the main mechanism for the eviction of H2AZ after its job has been done, which is to be there for the transcription machinery when it is just starting its work.
So for the single molecule dynamics work, I guess it was when you switched places from the NIH to Johns Hopkins University. So what was the motivation for this change in location? Was it because the Johns Hopkins University was a better place to do the single molecule dynamics work?
Well, a number of reasons. I mean, the NIH has been a wonderful place to work, and it continues to be so. I was there for 30 years. We discovered, footprinting of HSF1. We discovered NURF, chromatin remodeling activities there. We discovered SWR1, the histone exchange, H2AZ exchange enzyme there. After 30 years, this is now in 2012, I realized that if we really wanted to understand mechanism, the kind of biochemistry that we were doing in test tubes was basically not up to the task of revealing kinetics. And the reason is that, test tube reactions are what we call ensemble reactions. Every molecule has its own idea about when it should do its thing. And when you have, millions of molecules in a test tube, you end up with a population average. But a lot of these reactions, as I already alluded to, just take a few seconds. How on earth are we going to study kinetics when the reaction is over in a few seconds using test tubes? And so the way out of the problem was basically to take advantage of those great advances in single molecule imaging, in which you could look at molecules one by one. And so instead of using a stopwatch, you add the ATP and you wait, and you take time points. When you do single molecule imaging experiments one by one, where the whole ensemble of molecules are being observed one by one under the microscope together, you can throw away your stopwatch. And you just simply time your movies, as it were, based on the molecular behaviors of the individual molecules in your field of view. And so I had to basically relearn, go back to school, as it were. I took a 4-year, quote unquote, sabbatical at the Howard Hughes Medical Institute, the Genelia Research Campus there, which was about an hour from the NIH where I was. And opportunity arose that I could basically relocate and go back to the bench, or at least try to go back to the bench, and learn single molecule imaging biophysics. And it was absolutely crucial that I had that sort of block of time, which is unusual, in fact, unheard of, to do a 4-year sabbatical. But that's basically what I did. I went first alone, and then 2 other colleagues came from the NIH with me. The NIH operation, thanks to my supervisors there, allowed the operation to slowly wind down while I was winding up the single molecule imaging at Genelia. And the colleagues that I had, Maxime Dahan and Xavier Darzac, 2 young colleagues, biophysics people, and then other colleagues, senior colleagues, Bob Tijan and Robert Singer, and then the great advances in fluorophore chemistry, colleague Luc Levis was there, and of course Eric Betzig had invented super resolution microscopy. Basically allowed us to, look at these histone exchange reactions and chromatin remodeling interactions in live cells, one molecule at a time. And so after 4 years of that, we basically learned how to do it, and then we needed to reestablish our lab. And so the opportunity to come to Hopkins came up, and that's what we did. We moved from Genelia to Hopkins. Again, initially, 1, 2, 3 people, and now we have the usual lab of about 10 to 12 postdocs, grad students, a lot of undergrads as well, which we didn't have at the NIH. Uh-huh. Nor graduate students. So it's a different experience. So we're still in business. We're really excited about what we're doing, revisiting a lot of these traditional classical biochemical reactions, but at the single molecule level, where we can look at, reaction intermediates that would be hidden in the in testing reactions. But that reveals themselves at the single molecule level, and that only lasts for a few seconds. And so there's a lot to be learned, from a mechanistic point of view. And I think that, in fact, that one of the reasons why I decided to go to Hopkins was really to train new sort of students and postdocs in single molecule approaches to classical problems in chromatin and transcription biology.
So this already answered my next questions, which would have been, what is it that you are currently working on and will be focusing on for the next 5 years? So that's done. Check. So for the last 44 minutes, we have been on a journey through your scientific career. I'm sure we missed some important papers, but would you like to add something?
Oh, I think you've covered a lot. The DNase hypersensitive part site was really what got us started. And then if you dig into that, we ended up with transcription factors like the heat shock transcription factor, and that's still an active area of research. And then, of course, the remodeling enzymes that cooperate to basically mobilize nucleosomes around we did some work on histone variant, the other histone variant, which is a centromeric histone variant, SEMP-A, in which we identified a special chaperone for SEMP-A. We initially got into that partly because of the experience with H2AZ, where we discovered a sort of a uncharacterized member of the remodeling family. We didn't know what we were going to see when we looked at it, what is called a SEMP-A in humans, or CSE4 in yeast, a centromeric histone. But that was also a very, interesting and useful experience. That there's a special chaperone, and there's a whole field of histone chaperones that other people are working hard on. That's very interesting as well. But I think you've pretty much covered it all. And what we're now doing very much, as you say, we're really moving back to single molecule approaches on the classic problems of chromatin and transcription biology, but also paying a close attention to genomics and proteomics. And I think one of the great things about the field is the application of genomic approaches, this seq or that seq, which has given us a much more global view of what's going on in chromatin on the epigenomic landscape. And there are, there's 1,600 human transcription factors. And there are at least 24 or 36 sort of chromatin remodeling enzymes. I didn't even bring up the histone modification enzymes. So that's yet another, several dozen enzymes. And those are, we're starting to look at those as well at the single molecule level, because they all work together, right? I mean, it's histone acetylation enzymes, modification enzymes, demodification enzymes. And one of the things we're currently doing, which I'm excited about, is to follow up on a very early collaboration with David Alice's lab, where David called me one day and said, "Oh, we've been studying this histone modification, and looking for histone acetylation. That is, looking for things that bind to it, and picked up NURF as one of the main binders of acetylated histones." And so that led to a Nature paper. Joanna Wysowska was the senior author of that paper in David's lab. That was a very productive collaboration. It was exciting. And of course, it turns out that the remodeling enzymes invariably will have histone reader subunits. So the NURF enzyme has PhD fingers, has bromodomains, and the SWR1 enzyme has a bromodomain and a Yeats domain. So there are two subunits of the SWR1 histone exchanger that recognize acetylated histones. And in work that has been submitted and is under review currently, we sort of dissected out what's the role of the histone reader domains on histone exchange activity. And it turns out that the enzyme binds with higher affinity to acetylated histones, almost to the level of the DNA binding components. So all of these enzymes will have DNA binding components and histone modification binding components. And they put together in one enzyme multi-subunit complex will confer on the enzyme the ability, in the case of SWR1, to recognize the accessible DNA in the nucleosome-free region. So that's one approach to recruitment, is basically having accessible DNA for the DNA binding components. And there's a sensor subunit of SWR1 called SWIC3 that is responsible for directing the enzyme to the nucleosome-free region, to the accessible chromatin. But then these other subunits, BDF1 and YAF9, which have the bromodomain and the Yeats domain, also participate in the recruitment of the enzyme, but in different ways than the DNA binding component. So in this case, they bind to the acetylated histones in the +1 nucleosome, and the enzyme then combines DNA binding mechanism with histone acetylation binding mechanisms together to target it to the +1 nucleosome, because the +1 nucleosome is known to be enriched for histone acetylation and other histone modifications. So we have, and this is again work that is currently under review, but I think it gives us a much more comprehensive view of how these enzymes are targeted to where it matters most, which is, to the +1 nucleosome, because that's where you have to get the PIC connected to. And because we're doing single molecule imaging, we can measure the diffusion behaviors of these enzymes. And what it turns out is that there's diffusion in 3 dimensions in the nucleoplasm. We all know that. But surprisingly, there's a lot of one-dimensional diffusion, because when you create chromatin accessibility, you basically have a stretch of nucleosome-free DNA. Not very long, maybe about 150, 200 base pairs. But almost everything that we see is diffusing in 1 dimensions along that couple hundred base pair region. And where it goes, you can diffuse either to the left or to the right. If you go to the left, you hit the -1 nucleosome. If you go to the right, you hit the +1 nucleosome. And if the +1 nucleosome is enriched for histone acetylation, then one-dimensional diffusion becomes biased towards the +1. And so that's how we think, and we have evidence for this in this paper being under review, that the enzyme initially is bound to the nucleosome-free DNA anywhere along the DNA, and it is free to diffuse in 1 dimension. But if it's acetylated in the +1, then it gets captured and retained, what we call confined diffusion at the +1 location. So what I'm very excited about is really studying from bringing up the whole idea of confined diffusion in 1 dimension, also in 3 dimensions. There's confined diffusion. You probably know that protein clustering, phase transitions, phase separation into condensates is very fashionable these days in the nucleus for proteins that apparently are involved in chromatin biology and transcription. That's one extreme. And the other is that you don't necessarily need to undergo a phase transition. You can just have confined diffusion, and that's good enough to increase the kinetics of association by several fold. So I think there's going to be a spectrum of interactions on one extreme, phase transitions and droplets, and the other extreme in simple confined diffusion. They both work. Mm-hmm. I think the challenge is to keep an open mind in any reaction in the nucleus, and see exactly what is going on for the proteins that we're studying and for the processes that we're studying, because the tools are now available. Single molecule imaging tools are now available that will be able to distinguish between these 2 extremes in protein compartmentalization and protein diffusion.
So my last question, and this has been a very fun question for me to ask because it's always nice to hear people's opinions. So what is your personal definition of epigenetics?
Oh, I remember those days when there were huge debates about epigenetics. There's a classic definition, right? That you inherit a state which is not based on the DNA per se. I would prefer to just to use the word epigenomic as opposed to epigenetics. And what it means is that the DNA has a lot of things happen to it. It has modifications. It has histones bound to it. The histones are modified. There are transcription factors bound to the DNA. Any one of these can be, and of course, there's RNA as well, that are associated with chromatin, and potentially, and in some cases, with regulatory functions. Any one of these can be inherited, quote unquote. That is to say, it is carried on during cell division from the mother cell to the daughter cell. And so, I prefer the broader view of all of these things are quote unquote epigenetic or epigenomic in a sense, and not get too hung up on words, but more importantly on mechanism. And I think what I really want people to think about is just think about the mechanism. Don't worry about the words so much.
Yeah, again, a very interesting answer indeed. So thank you, Carl, for your time and for being on the show.
Thank you so much. I look forward to listening to your other speakers and maybe eventually even to myself.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.